Okay, I will start my talk on Trichomonasvirus virus. The title is A Parasite of Parasitic Protozoa. Actually, this is my work for the past 3 years. Before my talk, I will spend a few seconds to give a brief introduction on Trichomonas and Trichomonasvirus vaginalis. As you see from this movie, Trichomonas is a very lovely flagellate of the human urogenital tract. This parasite was discovered about 180 years ago, but the importance of Trichomonas was not yet understood until the 1990s. This is a report from the WHO about the global impact of sexually transmitted diseases. As you can see here, for each year, the case of Trichomonas is about 170 million, which is about 70% of the global sexually transmitted disease. So, I mean, Trichomonas is the most important neglected sexually transmitted disease. As I mentioned before, Trichomonas vaginalis is a parasite of the urogenital tract. So obviously, the only way to transmit Trichomonas vaginalis is by sexual activities. The life cycle of Trichomonas vaginalis have 3 different stages. In the human urogenital tract, it will exist as a superzoa. But when it attached to the vaginal epithelial cell or the prostate epithelial cell, it will turn to an amoeba form. If the environment is not suitable for cell division, it will turn into a pseudocyst. So, as I mentioned before, most of the Trichomonas are transmitted by sexual activities, but there has been reported some transmission between female sex partners and in newborns, some with pulmonary trichomoniasis. How to prevent this infection? Obviously, is simple. Just use condom for safe sex. But obviously, most people don't want to prevent trichomoniasis. If we look at the clinical symptoms of trichomoniasis, it's quite interesting because Trichomonas vaginalis is a kind of parasite that displays sex discrimination. In men, about 90% of the infected males are asymptomatic. In females, about half of those patients, they will have symptoms variation from sore cervix to vaginal ephemeral. And there has been some reports that female patients infected with Trichomonas, they will have low preterm no birth, infant birth, or the risk of HIV infections. Start from the 1960s, there's quite lots of reports on the pathogenesis of Trichomonas. As we can see here, the electron microscopy of Trichomonas, it says the parasite actually can destroy the vaginal epithelium cells. Cumulated experimental data have summarized the virulence factors of Trichomonas into different categories. The first one is the environmental factors. As we know, if the parasite is inhibited in the female vagina, because the menstruation cycle is constantly change the microenvironment of the vagina. So the iron, glucose, or other factors in the vagina, actually it will control the expression of virulence factors. The most important thing is the iron concentration in the vagina. Why is that important? Because Trichomonas vaginalis, it don't have a mitochondria. Instead, it uses a mitochondria-like organelle called hydrogelism for electron transmission. And the transmission is almost displayed by or carried by iron-cooperated proteins. Other virulence factors, including the excreted products from the Trichomonas vaginalis, there's a several reports mentioned that these ESPs actually can induce immune response in the human vaginal cells. The next slide shows is an interesting thing is because Trichomonas vaginalis is a protozoa, but in Trichomonas there is also a parasite, what we call the Trichomonas vaginalis virus. Trichomonas vaginalis is a double-strand RNA virus, and this virus was discovered about 20 years ago. There has been reports showing that early in the 90s that this virus can upregulate the expression of immunogen called P270. And other reports also says that this double-strand RNA virus actually have effects on the growth kinetics of Trichomonas vaginalis. And the most important thing is this virus have several different subspecies. And a recent report in Postnatal Tropical Medicine says that if a Trichomonas virus was infected by the virus, especially those type 1 and type 2, they are more sensitive to metronidazole, I mean, the drug that we use to treat Trichomonas vaginalis. So here comes a very important thing is what is Trichomonas vaginalis virus? So Trichomonas virus belongs to the family Totivirus. As this slide shows, it's a double-strand RNA virus, and it has a genome size of 4.6 to 4.9 kb. And in the virus genome, they only encode two different proteins, the capsid protein and the polymerase proteins. These proteins actually is quite interesting because at the C-terminal end of the capsid protein, there will be a frame shift to produce another overlapping protein called the polymerase 1. Talking about the parasite of the parasite, Trichomonas virus is not only the virus identified in protozoan. There's also like in Leishmania, we have the Leishmania virus. They have two different subtypes, virus 2 and virus 1. And in Giardia, we also have the Giardia lambda virus called Giardia virus. Also in other three different like Acanthamoeba, they have a huge mega virus. In Trichomonas vaginalis, up to March this year, we also always said that we have 4 different subtypes. But and this year, Max Neighbors of the Harvard University present a new report. They said they use NGS RNA sequencing. They identify a new species of Trichomonas virus. So up to now, we have 5 subspecies of TBV, what we call the TBV 1, 2, 3, 4, and 5. Based on the phylogenetic analysis, as we can see here, TBV5 and TBV2, they are more closely related, and also TBV3 and TBV4. And it looks like TBV1 is the progeny of all the other TBV. The next slide shows something about the exosome. So as we know, TBV can only be transmitted by vertical transmissions. It means that they will not cause lysis of the cell, or they will not release to outside the cell. Why I started to study exosomes? Because there's lots of paper reported that the exosomes from those cells infected with TBV or they have a different effects on different cells. As we can see here, it shows that the small extracellular vesicle from TBV plus and TBV minus cells, they have different effects on the host immune response. In this case, they use NF-κB and IL-X as an example to demonstrate that the virus, the exosomes from TBV plus cells have different effects on this lymphocyte expression. And also in this paper, they also demonstrated that the cells with TBV or without TBV, they have a different response to the response. They can induce different immunological response in host cells. So here comes the important part of my talk. Up to now, we have 3 different players. The parasite, Trichomonasvirus vaginalis, the parasite of the parasite, Trichomonasvirus, and exosome. So the first question I want to ask is, if TBV have 5 different subtypes, is it possible that in each isolate they have co-infection of different viral subtypes? And the question 2 is, I mean, there's a lot of papers demonstrated that exosomes from TBV plus or TBV minus cells, they have different effects on the whole cells. So is it possible because the exosome contain TBV? Or I have to ask is it possible that exosome can play a role on TBV transmission? The third question is, if exosome plays a role on TBV transmission, is it possible I use an exosome to induce TBV into other cells? So I will start from this wonderful experiment data. This data shows that I use RNA sequence, NGS RNA sequencing, to detect the virus in Trichomonas isolate. The isolate that I use is a reference isolate called ATCC5014X. Based on the mapping of RNA sequence to the reference sequence, as you can see here, this isolate has 3 kinds of TBV. They are TBV1, TBV2, and TBV3. Based on the total read counts, the major virus in this isolate is TBV2, followed by TBV1 and TBV3. So the next step is, I have RNA-seq data from my lab. We have about more than 150 RNA-seq data. So we start to we look at all those isolates that we have to see if they have TBV in different isolates. So this figure only shows this is the data set that we generated from RNA sequencing, and this is the name of the isolate, and the third column is the number of TBV identified in different isolates. So as we can see here, some isolates do not have any TBV. And quite interesting thing is, these isolates actually, they have been experimentally verified. These isolates, they are highly resistant to metronidazole. So based on this one, I mean, we can maybe imply that TBV plays a role on drug resistance. And some isolate, they will have only 1 TBV to 2 TBV co-infection. And interesting thing is, for this isolate NYCD15, it has 5 different virus subtypes altogether. And as I mentioned before, TBV5 is a newly isolated identified TBV. Based on this chart, it's quite interesting because you can see here, I mean, all those clones have TBV5. Actually, they are collected from New York City. So I mean, TBV5 may be a New York City specific TBV. Okay. The next step is, I mean, in our previous study, we already shown that, I mean, some isolate, they will have more than 1 TBV. So the next experiment we are going to do is, we need to demonstrate whether those exosomes secreted from these isolates, they also contain TBV. So what we are going to do is, we purify exosome. I mean, it's a quite difficult work from 6 different isolates, and then we use LC mass to identify whether there is a capsid protein in these exosomes. After we're doing the experiments, and then the next step we are going to do is, I mean, for LC mass, we can only determine the total population of TBV. But we also are interested in whether all the cells in a single isolate, they have the same TBV distribution. So we will use 2 different methods to demonstrate this one. The first one is clonal selection. It means we single clones from different isolate and then use our RT-PCR to demonstrate the TBV concentration of each cell. The next experiment is, I mean, it's quite heavy, and we use a a new technology called single-cell RNA sequencing to check whether single cells have the same TBV distribution. So here goes on our results. The first slide shows that we isolate the exosome, purified exosome from 6 different isolates. From ATCC026, I mean, these all are reference strands. This slide shows the results of our LC mass. The first thing is, I mean, in the first slide, we only demonstrated that we have the exosome because of their size and their classical cup-shaped form. In this slide, we need to make sure that these exosomes are really exosomes isolated from the tumors. So in this slide, we use the tetraspanin. Tetraspanin is a classical marker for, I mean, almost exosomes from all different species. From here, here it shows that all the exosomes, they have a tetraspanin on the protein extracts. So the next step is we are trying to use LC mass to identify whether there is virus in the exosome. This is only an example. I use 2 data. The first one is the T1 isolate. T1 isolate is, in our previous study, demonstrated that it has only one virus, that is TBV1. So, I mean, our LC mass data clearly shows that, I mean, the exosomes from T1 also have the TBV virus. Another example is the isolate ATCC50148. Based on our RNA sequencing studies, this virus should have 3 different TBV. So the LC mass showed the similar results. And from the LC mass data, we can identify the capsid protein from TBV1, TBV3, and TBV2. Okay, so the next step that will be interesting is we already know that in a single isolate, they can be co-infected with 2 or more TBV. So an example is the isolate PRA9H. I mean, it's also called G3. In G3, we use 2 different technologies show that the G3 have 2 different TBV. They are TBV2 and TBV3. So this is the result of the NGS data, and this is the result of our RT-PCR data. So what we're going to do is we are trying to grow the cells in a semi-solid medium instead of a liquid medium. And then we pick single colony from here and then extract the RNA and then use RT-PCR to check all those individual colonies. The result is here. We pick 48 single colonies from this plate and then extract the RNA and then we do RT-PCR. So in here, it's quite interesting because if we use RNA-seq or RT-PCR or even LC mass, we show that this isolate contains TBV2 and TBV3. But in fact, if we look at all single cells, we can see here is, I mean, they are not homogeneous because in this case, we pick 48 single colonies and among them, 36 have both TBV and 2 and 3. And 50% have only TBV2 and 6% have only TBV3. And 4% actually they are virus-free. So this is the first report to demonstrate even if you have TBV in a single cell, all the cells, they are not homogeneous distributed with TBV. Okay, so the next step is, we are trying to use another isolate, ATCC50148. This isolate, as I mentioned before, it has 3 different viruses, TBV1, TBV2, and TBV3. And this is the RNA-seq data. So what we have done is we use a new technology called single-cell RNA sequencing. In this technology, we use a cell separator to isolate single cells and then extract RNA from each single cell and use the RNA to do RNA sequencing. Oh, okay, this is the result. After we doing the sequencing, we can use the marker or the adapter with different sequence to identify whether they have TBV1, TBV2, or TBV3. So if we overlap these figures, we can have different kinds of combination. So the result is here. I mean, if you look at this isolate, as mentioned, it has 3 different TBV. Okay, but in fact, about 50% only have one virus, TBV1, 80% have TBV2, and 2% have TBV3. And there's a different combination. Some cells have only TBV1 or TBV2. Some cells have TBV1 and 3. Some cells have TBV2. And about 60%, so actually they are TBV3 or TBV minus. So based on this experimental data, we came into a very interesting conclusion is if we look at the TBV, we demonstrated a single isolate can be co-infected with more than one TBV. And this TBV actually, they can be transmitted through exosome. So what we call the exosome-mediated infection. And in these exosomes, they will have a TBV of different combinations. As I mentioned in the previous experiment, actually, I mean, in a single isolate, even they co-infected with different TBV, the distribution in each single cell is different. So our next step is, if TBV is important in drug resistance, as I mentioned before, those highly resistant cells, usually they don't have any TBV. So at this stage, I mean, we are trying to infect those TBV-free cells with TBV and test their drug resistance, whether that depends on the TBV or not. Okay, so this is the end of my talk. I think it's about time to stop now. I would like to spend all my lab mates. I mean, this photo is quite interesting because everybody is wearing masks for the dinner because COVID is still pandemic in Taiwan. Okay, thank you very much.